Hi everyone. So I'm Dinesh. Thank you for the introduction. So I'm a master's student at Department of Biotech, Indian Institute of Technology, Madras. So today my talk is going to be about a protocol for use for studying metabolic interactions in microbial communities using graph-based approaches. So this is going to be the outline. So we'll start with the motivation, and then we'll go into a little bit of background context, and then we'll discuss about the main protocol, and then few results that we got from the protocol, and we'll end with summary. Okay, so the main why we undertook this project is because we wanted to study microbiome. We know, I think everyone here would know the importance of microbiome. So microbiome, so studying microbiome is a bit difficult because they are very complex system, and then in order to study them we need to like understand how they interact with each other in a microbiome. So the difficulty here is to replicate them in laboratories, which is not very easy. So there is need for computational approaches to study microbiome. So most of the just a second. Okay. Yeah, so most of the computational approaches that work with something called genome scale metabolic models. Um, um, uh, one of the problem here is to we need to curate these models in order to use them. But is there a way to come up with a computational method which uses only no already available information? The solution would be to come up with a method that uses a uh, available information, which is the genome information. So we, thanks to the high throughput sequencing technology, we have a lot of genome information about a lot of microbes. So and then we need a method that has to be feasible to work with a large community data. So because microbiome are like they have a lot of members, lot of microbes form a microbiome. So the method that we need has to work with large data. So the plan here is to work with something called as metabolic networks, more specifically genome scale metabolic networks. So these are nothing but bipartite networks used to represent the metabolism of any organism. This kind of maps the whole topology of the metabolic reactions happening in any organism. So they can be constructed for any organism given we have the genome. So here in this protocol, we are going to use a special Python package called MedQuest for working with these metabolic networks. So what is special about this MedQuest? So this MedQuest requires only this topology of metabolic networks, nothing else. And then it is scalable to large metabolic networks, and then we can use so it kind of deals with all the multiple alternate routes that are present in any given metabolic networks. So it also kind of handles all the different components that are present in a metabolic network. Metabolic network would have something like export reactions, reversible reactions, irreversible reactions, internal reactions, external reactions. So the method has to handle all these different components when we are studying a microbiome. So which MedQuest does. Okay, so now that we have some resources, but what is studying a microbiome? Like how do we study a microbiome? Studying a microbiome is basically studying how microbes interact with each other. So microbiome is composed of a lot of different microbes. So if we kind of try to understand how they interact with each other in the microbiome, we would kind of get to know the dynamics of the microbiome, like what happens. So what can these interactions tell us? So it can tell us possible interaction patterns, these how these interaction patterns changes with the environment when this microbiome subjected to different kinds of environment, how this interaction pattern changes, what are the possible metabolic exchanges that can happen, and the dependence of one microbe on the other in the microbiome, metabolic capabilities of microbes in the community, and then how it affects the community. And it will also help us studying these interactions will also help us um, to come up with some hypothesis about understanding the dynamics of microbiome that we are going to study. So okay, so all right, so now let's get into the protocol. So first step in this protocol is to construct metabolic networks. Here, so we'll start with genome data, and then we use online tools like KBase and CARMI to construct these genome scale metabolic models. So and then from these models, we use our package called MedQuest to construct metabolic networks. Metabolic networks will be constructed for individual microbes, for microbial pairs, and as well as for higher order communities, that is like communities with more than two microbes. So once these networks are constructed, the next step is predicting metabolic exchanges. So how we do that is, so we have a set of metabolites that will represent the environment in which these microbiome, this target microbiome, lives in. So with this, when we simulate this metabolic network we have in this metabolic environment, which we'll do with this package called MedQuest, what we'll get to know is that we'll get to know the different reactions that are active and the reactions that are inactive in this given environment. So okay, what does this tell us? So when we looked at my metabolic networks that are like individual metabolic networks versus the metabolic networks that are community, we see that this reactions, that inactive reactions, are very less in when the microbes are in community. So that is because of the presence of exchanges happening between microbes in community. So by looking at the reactions that are relieved, or the reactions that are active in a community which were initially inactive when present in individual microbe, would let us know what are the metabolic exchanges that happened, that led to the active relieving of this inactive reaction. Okay, so the next step is predicting metabolic dependencies. So this the concept we were discussing earlier in the slide, the relieving of reactions is no directly tells about the metabolic dependency of one microbe on the other. So this says that we need another microbe to activate the, I mean, another microbe to no activate the inactive reactions. So these metabolic dependencies, we quantify these metabolic dependencies using two different scores called metabolic support index and community support index. So basically this score tells us about the increase in the metabolic capability of microbe in while in community versus while present individually. So calculating this, so metabolic support index is for microbial pairs and community support index is for higher order communities. Studying these different scores, determining these different scores will help us understand possible interaction patterns that might happen, that might occur in a community. And uh, so in case of higher order community, we'll also get to know something called as unique contributors. So unique contributors are those microbes when they have like some special unique metabolic capability. When you remove these microbes from the community, this the whole community will lose that particular metabolic capability. So finding these unique contributors, finding the interaction patterns of microbes will help us, will tell us a lot about the microbiome itself. So the next step is predicting metabolic capabilities. So we were talking about simulating metabolic networks, right? Like getting to know what reactions are active and what reactions are inactive in a given environment. This will also tell us about the metabolic capability. So what metabolites can be produced, what metabolites cannot be produced by a certain microbe for in a given environment. So summary, so these four things is a form of this protocol that we are talking about. like quick summary of what we were discussing is, so we had metabolic networks from which we tried to predict metabolic dependencies using this course called MSI and CSI, and then we were trying to predict possible metabolic exchanges that might happen, and then we were also trying to look at unique contributors that are present in the community. So this is about the whole protocol. So with this method protocol, we study different microbiomes, microbial data sets, hydrothermal vent data set, and ocular microbiome data set. So we'll see some interesting results that we got. First, with the hydrothermal vent. Hydrothermal vents are the small volcanoes that are present on the surface of ocean near to the earth core. So this is a very unique environment, and it has very unique set of microbes present there. So the data we had is about we had like 98 metagenomically assembled genomes of 90 different microbes. This had both archaea and bacteria. So we were a bit interested in studying how archaea interacted with bacteria. So this particular figure here shows the interaction pattern of archaea called Paseurchiota. So you can see that all the arrows are going towards Paseurchiota, which means that this Paseurchiota has an has the potential to get a lot of support from other microbes, while you can see only one arrow going from Paseurchiota to other microbes, which says that it has very less potential to support other microbes. So this might tell us about two things about this particular archaea. Uh, this this um, this is very dependent on other microbes while in this environment, and it has very less metabolic capability in this environment. So we were also able to capture the change in interaction pattern when the environment changes. We kind of used four different media conditions while studying this microbiome. So we were able to capture how the interaction pattern differs when the environment changes. So okay, so this is about hydrothermal vent. So next, let's see something about ocular microbiome. So here in this data, we had two different communities. One was healthy community, and the other one was bacterial keratosis infected community. See, we wanted to see how these two communities differ. What we can get to know about this two community using our method. So first, in this slide, the network here shows this is the network of healthy community, the interaction pattern of healthy community. Both are like both are healthy community, but no, in different conditions. The first one is in healthy condition, while the second one is in the infected condition. You can see that there are a lot of edges in the healthy condition. The each edge corresponds to an interaction between a microbe, between microbes, and you can see that there's a lot of edges. That means there's a lot of interactions in healthy condition, while there's very less amount of interaction in infected condition. um, this was something we captured. We were able to see using this method. And the other one was so this is a very special community called combined community. What we did here is we combined both the healthy and the infected microbes. So we wanted to see how healthy microbes interacted with the infected microbes. So this is um, the reason we did this is because we wanted to in this transition stage, when a healthy community becomes a infected community, there'll be interaction between these two microbial communities. So this studying combined community would let us know about this transition stage. So here again you see the combined community in two different conditions, healthy and as well as from infected. In healthy condition, you can see that there's a lot of interactions, while in infected, there's very less interactions are retained. The interaction retained here are mostly from between infected microbes. So right, so to summarize everything, we first introduced a protocol for studying microbial interactions, and then demonstrated the working of this protocol. And then we try to use this protocol to identify some interesting results about the two different microbiomes. And as a future work, we can also use this method to study microbial assembly, or we can extend, we can perform other constraint based modeling to fortify the results that we get from this method. And of course there are some limitations to it. The metabolic networks are only as good as the genome quality we have, so it is important to have a good quality genome data. And this method is dependent on the metabolite environment that we supply. So it is important to have proper representation of the metabolite environment to get accurate results. So that's about my presentation, and I would like to thank my advisor, Dr. Karthik Raman, for helping me with this project, and Dr. Karthik Anandaraman for our collaborator for providing the hydrothermal vent data, and collaborator Dr. Shivaji for providing the ocular microbiome data. And I thank my lab mates for their support. And I thank Ministry of Education India for supporting my master studies, and ISMB Travel Gan for allowing me to attend this conference. Thank you. Be happy to take. So the slide designs I used are from Slides Go and Slide Carnival. And thank you.
Any question for Dinesh?
Thank you for this wonderful talk. I have a question about what you mentioned on the summary slide is about the representation of the metabolic environment. So you showed several environments where these microbes were living. So how well known are these media compositions, and where do you get information about what are the compounds and what the concentrations are? Do you have any data for that? That would be very interesting, and how you incorporate that medium information.
So in case of hydrothermal vent, we had, so our collaborator gave us some media, so information about the different media composition. So in that case, we used those. In case of ocular, we used something called as so we use this package Cobra Pi, where we'll be able to get minimal media. So this is based on the concept that what the metabolite environment, this depends on the microbial composition that there is. For example, like for a healthy community, we have 13 microbes. So we'll be finding minimal media that is required for the growth of every microbe, and then we'll be collating that, and then we'll be using that as the healthy condition. The same goes for infected.
Okay, that means you make a minimum medium for every bacteria species in that community, and then you make kind of one medium that has all the compounds that they all have in their minimal.
Yes.
Okay. Thank you.
Thank you. Thank you.
Okay, I think there's a question in the chat. So the question is, did you compare your tools with other tools for human communities, such as MICOM, ISMB, etc.?
No. So MICOM would require, so we only had draft genome scale metabolic models, and using MICOM would require us to have curated genome scale metabolic model to get some accurate results. So we kind of didn't use that, but we did some Steadicom. So we used this another tool called Steadicom for ocular microbiome, where we try to see what are the type of interaction, like whether it is parasitism or it's mutualism, those kind of things. So the thing here is that so the interaction captured here will only be positive interaction in sense like so if Steadicom shows there is parasitism, in which case like one microbe is decreasing the growth of other microbe, that interaction will not be, we will not be able to capture it in MedQuest, because it's not essentially a metabolic exchange. It's either like a growth of one microbe stopping the growth of another microbe. It does not necessarily mean there is metabolic exchange happening between them.